For galvanic cells, it's possible to measure the cell voltage. Here we have a reaction between zinc and copper ions to make copper and zinc ions. So this could be zinc plus copper sulfate forms copper plus zinc sulfate. The zinc is getting oxidized, which it does very easily. The copper 2 plus is getting reduced, which it also does spontaneously. We can actually do this chemical reaction, and there's the basic electrochemical cell right there. There's your copper 2 plus solution. See how it's blue? And on this side, this is your oxidation side full of zinc with a zinc anode right there. And so the anode would be getting consumed. The cathode would be getting plated out with nice copper, and the ions would be transferring over via the salt bridge. The difference in the electrical potential can be measured, and you'd find that it's 1.1 volts. That would be the cell voltage. It's also called the electromotive force or the cell potential. And it requires that the concentration of the ions here are 1 molar and here 1 molar as well. We could draw this as a cell diagram where these single lines means that you have a phase boundary for zinc going to zinc 2 plus here, right? And as it does, it transfers electrons over, which will in turn turn the copper 2 plus into copper as it gains those 2 electrons. So we call those the phase boundaries. And we call the double lines the salt bridge, where the salts are getting transferred because we're forming copper ions here, and so we need some kind of cation to replace the sulfate ions that were formerly copper sulfate. And similarly here, we're making zinc 2 plus, so we need some anions that can travel into there. So that's the function of the salt bridge.